Hello and welcome back to the channel everyone. Follow us, subscribe to the channel and hit the bell icon for all the updates.
Hello and welcome back to channel Pharmacology Simplified. In today's video, I would be discussing about the anti-epileptic drugs, and before that I would be explaining a little bit in brief about the pathophysiology or how epilepsy is developed and how do we develop the therapeutic approaches for management of epilepsies. So the question arises, what are epilepsy? But before that, we have to answer one question, that what are seizures? So we can say that seizures are, these are a sudden alteration in the behavior due to unsynchronized and rapid firing of the neurons in a particular area of the brain that may involve only one hemisphere, or it may involve both the hemispheres of the brain. And seizures are like when there is the epilepsy is when there is unpredictable and very frequent occurrence of seizures that is called as epilepsy, right? So epilepsy is a group disorder where seizures can be precipitated without any forcing reasons, right? So the next thing is how do we classify seizures, or how do we classify epilepsies that are seen in humans, right? But still, like the pathophysiology or how the disease is developing, it is not very clear to all of us, and the therapeutic approaches that are being used they are not very pinpointed, or they are not very accurate. So the drugs that are given during epilepsies, they have a broader mechanism of action rather than on a specific target, right? So when we say that simple and complex, the seizures they are of two types, right? When we talk about seizures, they are of two types: simple seizures and complex seizures. The only difference between simple and complex seizures is that during the simple seizures, there is no loss of consciousness in the individual, while in the complex seizures, the consciousness of the individual is lost. So this was about the seizures, and if we see how we categorize, or how what are the classes of epilepsy, so there are two primary classes of the epilepsies. First one are your partial, and second one are the generalized epilepsies, right? In the partial epilepsies, what happens? There is involvement of only one hemisphere of the cerebrum, while in the generalized, it involves both the hemispheres. But in some cases, it has also been seen that partial seizures, they get converted into the generalized seizures, right? Like the electrical activity was initiated in one hemisphere, right? It was localized, but over a period of time, or what happens? It involves both the hemispheres of the brain, right? So, for example, if let us consider if this is the brain, right? It's a cut-away diagram. So, if this is the brain, then what is happening? Let's say we have separated into two hemispheres, left and right. So first the epilepsy, for example, it was developing here. For example, it was localized into this particular area. Then it would be called as a partial seizure. But if this involves both the hemispheres, right? If it involves both the hemispheres, now this will be called as generalized, right? So now if we see that what are the various types of epilepsies that we can observe in an individual. So first we categorize them into partial and generalized. Partial is like where there is involvement only one hemisphere of the brain, while in the generalized there is involvement of both the hemispheres of the brain, right? So now the classes are simple, complex and complex tonic-clonic, right? So in simple, what happens? The episode lasts only for 20 to 60 seconds. There is no loss of consciousness. This is the key feature that differentiates it from every other. There is jerking of extremities. Usually it happens like thumbs. The thumb or the fingers, they will start to jerk. They will show jerky motions. The motions will not be smooth. And paresthesia will be there. In the complex seizures, the complexity will increase. It will be shown by loss of consciousness. Jerking will be there and it would last for 30 seconds to up to 2 minutes. The episode can last for 30 seconds to 2 minutes. And in the next one, the complex tonic-clonic seizures, what happens? These are the simple seizures. Simple seizures, they result into tonic-clonic seizures. Like what is happening? They are developing into a full. They are moving into the generalized form. So the whole body will undergo into jerking motions. There will be jerking in whole of the body, right? And the episodes can last for 1 to 2 minutes, and it is shown by loss of consciousness, right? So complex tonic-clonic is just what we can say is a bad version of the partial seizure, right? A simple partial seizure. Next category is generalized. This will involve both hemispheres, right? It will involve both hemispheres. First are the absence seizures. I will explain the like how these seizures are developing. Absence seizures, they arise only in the thalamus, right? Which means they will not show, there will be no muscle jerking. Remember this. There will be no muscle jerking in absence seizures. What happens in absence seizures? The individual will keep on staring. The individual will go absolutely blank for some time. The episode lasts for approximately 20 seconds to 60 seconds or for a minute or so, and individual goes completely blank, right? There is no movement of any muscle, right? Myoclonic, and now you can also think like sometimes while studying we go blank, but please mind that they are not absence seizures, because it's like we don't just bother about anything, so we go blank. They are not absence seizures. And next is myoclonic seizures. They are strong jerking of the extremities, which means they will be involving your skeletal muscles, right? The impulses will be arising from the thalamus to your cortex. And shock-like movement, right? The movements will be very rapid. And next are tonic-clonic. Tonic-clonic, I have just explained what are tonic-clonic. Just the difference is that in the partial tonic-clonic seizures, myoclonic seizures, there was first they were starting from the simple partial, and now in this case there is no episode of simple partial. They are not preceded by a partial seizure. They will go full on, right? They will go full on into tonic-clonic. The individual will fall down, all the muscles will go contracting, they will shiver, right? Okay. Now let me explain you how these seizures they are developing. So now let us see what are the different types of epilepsies and how do they occur. Right? First, we have to understand that how our brain initiates a movement. Right? Whenever, like, I'm talking to you, I'm moving my hands, I'm writing something on the board. So this all is a very coordinated action that is being performed by my brain. Right? So the action starts. How it starts? Like, this is the cortex master. So whenever it starts to initiate a movement, what it will do? It will send a signal to the basal ganglia. You know the components of basal ganglia, like the stratum is the part of basal ganglia, the chordate nuclei, the substantia nigra, pars compacta. All these are the parts of the basal ganglia. They also basal ganglia plays a very important role in the development of Parkinson's as well. My next video. So cortex will send a signal to the basal ganglia. Basal ganglia will rectify it, right? And then it will send the signal back to the thalamus, right? And now this signal from the thalamus will be sent back to the cortex. And now the cortex will send the signal to the spinal cord. So here, if this is the spinal cord, so what is happening? This is the cortex, right? So from the cortex, the signal arises, and through the basal ganglia, it goes to the thalamus. And now the thalamus processes the signal and it sends it to the cortex again, and from this cortex it goes down to the spinal cord, right? And through the spinal nerves, right? We know the spinal nerves. Through the spinal nerves, it is acting onto the muscles, right? For example, this muscle is contracting, so the signal generates in the cortex. It goes through the basal ganglia to the thalamus, then again to the cortex, and then through the cortex, it comes down through the spinal cord and goes into this in my hand, right? And now this muscle will contract, right? Okay. So in case of partial seizure, for example, this is the site, right? This is the site where the neurons start to fire, and from this fire, I mean depolarize. And these neurons start to depolarize on their own without any reason. And when they depolarize, what they do? They start to send signals, right? Right? Now, what is happening? These nearby neurons, they are being affected by this. Now, what will happen? This will lead to development of a simple partial seizure, right? So, whatever the area of our body that is being regulated by this will be affected, and I mentioned that there would be slight jerking because it is restricted into a smaller area of the brain, so not a big part of the body is affected into this. So this is partial, and more or less as the form progresses, it converts into generalized. Now, in the generalized, I have to explain. There are two major: absence seizures and generalized tonic-clonic seizures, GTCS, right? So in the absence seizures, I told that they occur only in the thalamus. And remember, the thalamus has T-type calcium channels. T is for transient, which means they are slow channels. So what happens? For example, this is thalamus, and the neurons from the thalamus, they start to fire. And when these neurons will fire, they will fire into the areas, right? And thalamus is essentially plays a role in our cognition also, right? So what happens? But the signals, they are not that strong enough that they go to the cortex, which means that the signals that are arising from this thalamus, they are not going to the cortex. Hence, in absence seizures, there is no jerking of any of the muscles in the body, right? And in the GTCS, what is happening? The signal will arise in the cortex, right? The center would be cortex. The cortex will send signals to the thalamus. Thalamus will now send these signals to the cortex again. Again, these signals will be sent to the cortex, and from the cortex, they will go to the spinal cord. And when these signals will go to the spinal cord, there will be rapid twitching and movement of the muscles, right? So this is about your GTC. So these are the basic differences about the basic features of the epilepsies, different epilepsies. Now we will move ahead with the pharmacological approaches that we can use for managing epilepsy. So now here we will see that how these ions play a role in development of epilepsy, right? So the first, these two are the presynaptic neurons, and this is the excitatory neurotransmitter because it is working with glutamate, and this is the inhibitory neurotransmitter because it is releasing GABA. GABA is inhibitory neurotransmitter while glutamate is the excitatory neurotransmitter. So now whenever an impulse arrives, so these are your sodium channels, there will be influx of sodium. So this sodium influx will lead to depolarization. This leads to depolarization, and in response to this depolarization, these calcium channels will open and it will lead to influx of calcium inside the cell. Whenever there is influx of calcium inside the cell, it leads to membrane fluidization. Right? Membrane fluidization is the process where your membrane becomes more fluid, more liquid-like, so that this neurotransmitter, glutamate, this vesicle will fuse here. Right? This vesicle will fuse here and it will release the neurotransmitter. Now, this neurotransmitter will go and bind to its receptor, NMDA receptor and AMPA receptors. Right? These all are ligand-gated ion channels, and now they will lead to the influx of sodium and calcium. This will lead to sodium influx, and this will lead to calcium influx, and this will lead to sodium influx. As a result, both of these will lead to depolarization, and the impulse will propagate to the neuron. Okay, but during epilepsy, the process is not this simple. This process gets intensified, right? This process gets amplified. As a result, there is rapid firing of the neurons, right? There is very rapid firing of the neurons without any reason, right? And there is one more phenomena that is called as depolarization shift. Depolarization shift. So what is this depolarization shift? That when one neuron starts to depolarize, it takes up all the nearby neurons into the depolarization. So there is an area where all the neurons are very rapidly depolarizing, and the brain is not able to process all the information that is coming through firing of all these neurons, right? As a result, the brain shuts itself down, and there is loss of consciousness, and the epilepsy proceeds, right? So this is how the impulse transmit. This is the excitation part. And for inhibition, every process in our body that has started, it has to stop also, right? The feedback mechanisms that we have studied. So now what will happen for the inhibitory purpose? For example, during physiological condition, the impulse function has been over, it is over, and now this impulse conduction has to be inhibited. So this is the postsynaptic neuron. It has GABA chloride channel complex, and how this GABA chloride channel complex functions, I will be mentioning the link here in the description, so you can go and check how these GABA chloride channel functions, right? So there will be release of GABA, and GABA is synthesized from, surprisingly it is synthesized from glutamate, which is acting as a excitatory neurotransmitter in the brain. It is getting converted into GABA, right? So from glutamine amino acid, glutamate synthase is the enzyme that is responsible for synthesis of glutamate, and now glutamate decarboxylase is responsible for synthesis of GABA from glutamate. Now this GABA is released, and it goes and binds to the GABA chloride ion channel complex, and as a result, this binds to the chloride ion channel complex. There is influx of chloride. There is a rapid influx of chloride ions. Right? There is rapid influx of chloride ions, and this leads to hyperpolarization of the neuron. And when the neuron gets hyperpolarized, it stops conducting. I am also providing a link for neurotransmission videos that I have previously uploaded on the channel. So if you have any doubts regarding the process of neurotransmission, which is very important, you need to go and check out that video also, right? So now this is the whole setup where now we can modulate the targets and draw some drug therapies, right? So pause this video for a moment and start to think that where we can do the modulation in this whole setup so that epilepsy does not occur. If we see that there is an imbalance between the excitatory and inhibitory neurotransmitters, and what we have to do, we have to level them, right? So that excitation and inhibition both take place simultaneously, not simultaneously, but in accordance with each other, right? So what we have to do, we have to reduce the excitation in the brain, and we have to partially increase the inhibition in the brain. So for increasing the inhibition, what we will do, we will stimulate GABA, and for reducing excitation, we will do what we will do, we will inhibit sodium channels, we will inhibit calcium channels, and we will inhibit glutamate, right? how we are going to achieve this. For inhibiting your sodium channels, we have drug called phenytoin. For calcium channel inhibition, we have carbamazepine and valproate. We also use valproate here, right? Then, so we have taken care of these two. Now, in order to increase GABA activity, so after the action of GABA is complete, sorry, after the action of GABA is completed, what happens? This GABA is reuptaken back by GAT1, gamma amino transporter 1, and this GABA is acted upon by an enzyme called GABA transaminase, right? This converts into semi-succinylate SSA. So this GABA becomes useless. So now what we will do, what we can do, like we can inhibit this GAT1 so that there is more availability of GABA. We can inhibit this GABA transaminase so that this GABA does not get broken down. And when this GABA will not be broken down, what will happen? That we will have more GABA so that we can use for will be available for inhibiting, right? So now let us see that what are the drugs that we are going to use. For calcium channels, we can use ethosuximide. Ethosuximide is used in absence seizures. Right? So what we have done, we have inhibited these sodium channels, we have inhibited these calcium channels, right? Other drug, the most extensively used, valproate, it has several actions. Like I have already mentioned, valproate inhibits sodium, inhibits calcium, it stimulates the activity of glutamate synthase, but it inhibits GAT, right? So it is very essential, very useful, valproic acid. I think I would I would separately upload the notes of this anti-epileptic drugs on the community tab as well as on the Facebook page. So if we see, like these are the sites where we can utilize for the management of epilepsy, right? What we have done, we have reduced the excitation, right? We have reduced the excitation, and what we have done, we have increased the inhibition by increasing the activity of the GABA, right? So with this, I would like to end this video. If you have any questions or any suggestions, you can mention in the comment down below. If you have been able to learn anything out of this video, please make sure to subscribe to the channel, share the video as much as possible, and happy learning to all of us. Thank you very much. So now we will see like what are the epilepsies.
बेटा नाश्ता लगा दूं क्या?
अभी कर लूंगा थोड़ी देर में.
ठीक.
रिकॉर्ड कर लेंगे भाई साहब.